graph, we want to look at the nodal cell action potentials. And these are the cells of the specialized conducting system of the heart. The cells that initiate depolarization, the stimulus for the entire heart to both depolarize and then contract. And we're going to look specifically at our SA node cells, as these would be the cells that are the pacemaker of the heart. And the only reason they're the cells that are the pacemaker of the heart is that they are the fastest to spontaneously depolarize. In other words, the first domino to fall makes all the other dominoes fall. And so the SA node will fire this action potential. It will travel throughout the atria to the AV node. They'll then cross the fibrous skeleton and enter the septum, the interventricular septum of the ventricles, and then up the ventricular walls. And so we get this very ordered pattern of both depolarization to generate a very ordered pattern of contraction and pressure generation to drive blood flow through the chambers of the heart and out into our systemic and pulmonary circuits. Now, we look at the action potential phases up here, right? And we're going to label those down below. And we see that it's not a numerically sequenced numbering here. And that's because we don't have all the same phases that we have in the cardiomyocytes. So the numbering is a little bit odd. We have our cell membrane here, this representative double line here, that's going to represent the barrier across which our ions are moving through voltage-gated channels to generate this action potential. And then, of course, down here at the bottom, we have our action potential occurring over time. Now, in phase 4, we have the very important phase that generates the spontaneity of our pacemaker cells, these nodal cells, and that's referred to as the pre-potential phase. And a pre-potential phase is generated largely by, what's called funny current. I is the abbreviation for current and F for funny. And this is funny current, or designated as funny current, because these channels open, these voltage-gated channels open in response to an odd stimulus. They open in response to a repolarization, a more negative membrane potential. Most voltage-gated channels that we've seen so far in skeletal muscle or in neurons are depolarization gated. They open in response to the membrane potential becoming more positive. So these are a little bit funny. And so as our action potential ends over here, we're going to enter a new action potential to the right here in phase 4. Right? It's the repolarization that generates the opening of these funny channels and leads into our next action potential. So this is sort of a looped graph here. Now our funny current is carried by two ions through these funny channels. It's carried by sodium current and potassium current. And of course, sodium is going to travel from outside to inside, ECF to ICF, down its electrochemical gradient. Potassium is actually traveling in the opposite direction through the same channel. Now those tend to cancel each other out. However, you see that we are depolarizing during this pre-potential. That means that the sodium ion and the sodium flux is the dominant current during the funny current phase here, the pre-potential. This leads us to the threshold here of another voltage-gated channel. Right? It's going to open a voltage-gated calcium channel. This is a long-term calcium channel. There is a short-term or transient calcium channel that opens at the end of the funny current here, helping to add to the pre-potential. But more importantly, we reach the threshold of this voltage-gated calcium channel, a long-term calcium channel, that carries the depolarizing current of the action potential here. Now this is a little odd as well because normally depolarization is generated by sodium influx, not calcium influx, so it's a little bit different in these nodal cells. This depolarization, we completely depolarize these cells. This depolarization will end, and that will actually stimulate the opening of another channel, a voltage-gated potassium channel. Potassium, of course, would then leave the cell, right? ICF to ECF. This makes the membrane potential more negative again, which again drives us to, it repolarizes the cell, but it also initiates opening of the funny channels again over here. And that's absolutely essential. What it means is that each action potential of the nodal cells generates the next action potential. This is what generates that spontaneity of our heartbeat. We do not require neural or hormonal input to the SA node to generate our heartbeat. That is not to say there is not neural or hormonal input. That neural or hormonal input will modify our heart rate, but it does not generate it. When we have that kind of input, and honestly, the funny channels actually have another name. They're called HCN channels. This indicates that they are hyperpolarization activated, right? We just talked about that. When the membrane potential becomes more negative over here in phase 3, that opens the funny channels again. But they're also cyclic nucleotide gated. Well, cyclic AMP is a cyclic nucleotide, and this is an intracellular signaling molecule that's utilized by things like the sympathetic and parasympathetic divisions of the autonomic nervous system. And so we can either adjust the funny current up or down based upon the prevalence, the amount of this cyclic nucleotide, cyclic AMP. And so basically what's going to happen is we're going to alter the slope of this pre-potential phase, making us get to threshold sooner or making us get to threshold slower, right? Separating our action potentials in time to differing degrees, right? If we steepen the pre-potential phase, we have a faster heart rate, sympathetic output. If we decrease that slope through reduced funny current, we would of course have a slower heart rate. That would be indicative of parasympathetic output. And again, we'll talk more about the cardiodynamics a little bit later.